A history of shaping life, from breeding to breakthroughs. For millennia, humans have shaped the living world around them. They didn't realize it at the time, but they were engaging in genetic manipulation. Farmers and breeders selected plants and animals with desirable traits. These traits were passed down to offspring, leading to gradual changes over generations. This selective breeding, though rudimentary, laid the groundwork for our understanding of genetics. The discovery of DNA in the 1950s was a pivotal moment. This double helix structure held the blueprint of life, unlocking the secrets of heredity and variation. The understanding of DNA's structure and function opened up unprecedented possibilities. Scientists could now study the very code that dictated life's traits. This knowledge paved the way for a new era, the era of genetic engineering. No longer were we limited to the slow and imprecise methods of selective breeding. We now had the potential to directly alter the genetic makeup of organisms. The implications of this newfound power were vast. We could potentially engineer crops for higher yields, develop livestock resistant to diseases, and even treat human genetic disorders. The ability to directly manipulate the building blocks of life promised a future where we could rewrite the rules of biology itself. This marked the beginning of a scientific revolution, one that would forever change our relationship with the natural world. The journey from selective breeding to understanding DNA was long and arduous. Yet, it laid the foundation for the remarkable advancements in genetic engineering we see today. With each passing decade, we delve deeper into the intricacies of life, uncovering new secrets and pushing the boundaries of what's possible. The story of genetic engineering is still being written, and it's a story that holds immense promise for the future of humanity. The genesis of genetic engineering early milestones. The 1970s marked the dawn of genetic engineering. Scientists, armed with their newfound understanding of DNA, began experimenting with ways to manipulate it. Early efforts involved inserting genes into bacteria and animals, often with unpredictable results. These were the first tentative steps into a world of possibilities. The first major milestone came in 1974 with the creation of the first genetically modified animal, a mouse. This breakthrough demonstrated the feasibility of altering the germline of an organism, meaning the changes would be passed down to future generations. The 1980s saw the commercialization of genetic engineering. The first genetically modified product was a microbe engineered to absorb oil spills. This marked a turning point, showcasing the potential of this technology to address real-world problems. The agricultural industry soon followed suit, developing crops resistant to pests and herbicides. The world was on the cusp of a genetic revolution, one that promised to reshape agriculture and medicine. The first genetically modified food, the flavor Sava tomato, hit the market in 1994. This tomato, engineered for longer shelf life, was met with mixed reactions. While some embraced the potential benefits of GM foods, others expressed concerns about potential health and environmental risks. This debate continues today, highlighting the complex ethical and societal implications of genetic engineering. Despite the controversies, the field of genetic engineering continued to advance. Scientists developed new techniques for gene editing, each more precise and efficient than the last. However, the true game changer was yet to come. A revolutionary technology, first discovered in bacteria, would soon propel genetic engineering into an entirely new era. CRISPR, a revolution in gene editing. CRISPR, an acronym for clustered, regularly interspaced, short palindromic repeats, emerged as a revolutionary tool in genetic engineering. This system, originally discovered as a bacterial defense mechanism against viruses, allows scientists to edit genes with unprecedented precision and ease. Unlike previous gene editing technologies, CRISPR is relatively simple, inexpensive, and highly efficient, making it accessible to a wider range of researchers. CRISPR works like a pair of molecular scissors guided by a programmable RNA molecule. This RNA molecule can be designed to target specific DNA sequences within a cell. Once the target sequence is located, the CRISPR system cuts the DNA at that precise location. The cell's natural repair mechanisms then kick in, either disrupting the targeted gene or allowing for the insertion of new genetic material. The implications of this technology are profound. CRISPR has the potential to cure genetic diseases, develop new disease-resistant crops, and even enhance human capabilities. It's a tool with immense power, capable of rewriting the very code of life. However, with great power comes great responsibility, and the ethical considerations surrounding CRISPR are as significant as its scientific potential. The development of CRISPR has ushered in a new era of genetic engineering, one characterized by unprecedented precision and accessibility. It's a technology with the potential to solve some of humanity's most pressing challenges, but it also raises profound ethical questions that we must carefully consider as we navigate this uncharted territory. The CRISPR revolution is here, and its impact on the future is only beginning to unfold. Imagine a world where diseases like cystic fibrosis, sickle cell anemia, and even some forms of cancer could be effectively cured at the genetic level. With CRISPR technology, this isn't just a dream, it's becoming a reality. By precisely targeting and editing faulty genes, scientists are paving the way for groundbreaking treatments that could transform healthcare as we know it. But CRISPR's potential goes far beyond just curing genetic diseases. This revolutionary tool is being explored for applications in agriculture, where it could help create crops that are more resilient to pests and climate change. It's also being used in environmental science, potentially aiding in the fight against pollution by engineering organisms that can break down harmful substances. The possibilities are, in many ways, limitless. As we embrace the incredible potential of CRISPR, it's crucial to navigate this revolution responsibly. Ethical considerations must be at the forefront of our minds. Questions about the long-term impacts on the environment, the potential for unintended consequences, and the moral implications of genetic modification need to be thoughtfully addressed. It's a powerful tool, but with great power comes great responsibility. In recent years, we've seen remarkable advances in CRISPR technology. From more precise editing techniques to new delivery methods, the science is evolving at a rapid pace. Looking ahead, the road is filled with both challenges and opportunities. Continued research, ethical guidelines, and international collaboration will be key in harnessing CRISPR's full potential while ensuring it's used for the greater good.